So we already know what the parts of the eye are. They were part of our last lesson. We did our eye dissection. Now we need to take a look at sort of how your eye works in a bit more detail. So the first thing we have to look at in more detail are photoreceptors. So I already briefly mentioned what they are. Photoreceptors are sensory receptors that detect light. Photo means light. There are two types of photoreceptors that we'll mention. The first are called rods. Now over here, this is a picture of what rods and cones look like. The one that's on the right that looks more like a baseball bat, that's a rod. The density of rods decreases as the distance from the fovea increases. So the farther and farther you get away from the fovea, and if you look at someone's eye, the fovea is this spot here that says 0 degrees. It's directly across from the lens. It's where the light should be refracted when the lens refracts it. So the fovea is in the middle of the eye, and if you go around this side or around that side on the retina, so you get farther away, there are fewer and fewer rods. Now rods, the way they work, they have a protein called rhodopsin. They can detect small amounts of light. And so rods are really good for vision in low light, like at night. And so maybe you've noticed this, or maybe you will now that I tell you this, but at night it's harder to see color. And one of the reasons is you're mainly using rods, and they are for black and white vision. They do not detect different wavelengths of light and interpret as color. They are detecting light or no light, right? So either white light or black, no light. That's what rods do. Cones are concentrated on the fovea. That's really the only spot in the retina that you find a significant amount of cones. They have a protein called photopsin, so it's similar to the protein that's in rods. But cones, instead of detecting just light versus no light, detect different wavelengths of light. So even if you're not taking physics, hopefully you remember like grade 8 science, electromagnetic spectrum. Each color of light has a different wavelength. So red light is a different wavelength than purple light, which is a different wavelength than green light. So you have cones, and you actually have more than one type of cone, but that's beyond the scope of our course. Okay, so I'm not going to ask you to differentiate the different types of cones. Just know you actually have several different types of cones that each detect slightly different colors, and that's how you interpret color. Now this picture that's in the middle is trying to show you, compared to the fovea, how many rods and cones does a person have? So it's referring to this first picture. 0 degrees is the fovea. And then as you get an angle that's farther and farther away from that, the number of photoreceptors change. So if I look at 0 here, it's just the dotted line for cones. There are cones on the fovea. That's basically all there is. If I start moving in either direction, the number of rods is really high, and then it slowly decreases. So that's these lines here, as I move farther and farther away from the fovea. And then there is a spot, the blind spot. So notice here at about 20 degrees from the fovea, there's the optic nerve. So there's a spot where there are 0 photoreceptors, none. Not just a few or not very many, 0. Now, does anyone want to ask anything so far about rods and cones? On the picture, the cone is the one that's pointy, looks kind of like a cone. So we have these two different types of photoreceptors. So you probably wonder what on earth they do to help you detect light. So light will come into your eye from the front and make its way all the way to the retina at the back of the eye, where you have rods, which are shown in gray here, and cones. And you'll see, look, there's a green one, a red one, a blue one, because there are 3 different types of cones. That picture is just for show. It's not anything new or different. It's just a different type of illustration. Now, in rods, I said they had this protein called rhodopsin. It's a pigment. It's actually made up of two pieces, retinal, which is vitamin A, and opsin. In the dark, rods release a neurotransmitter, which inhibits nearby nerve cells. So when we talked about neurotransmitters, we said they could be excitatory and cause action potentials, or inhibitory and make the membrane hyperpolarized and make it so there wouldn't be an action potential. So in the dark, rods are releasing this steady stream of inhibitory neurotransmitter. When light is absorbed, that pigment, the protein, rhodopsin, divides. When that happens, we stop releasing the neurotransmitter. So I stopped releasing something inhibitory. It's like I took the brakes off. Now that I'm not releasing this inhibitory neurotransmitter, nerve impulses can pass through. So this is an unusual thing, right? Normally we want something to happen to cause something else to happen. Here, we're taking away the inhibition, and that's what's allowing the nerve impulse to start flowing. Now in cones, the events are similar, but the pigment is photopsin, not rhodopsin. So I didn't repeat the steps because it's a very similar sort of process. I also didn't name the specific neurotransmitter because I don't need you to know stuff in that much detail, and it's different for the cones and for the rods, and I don't want to gum up your brain with all this excess stuff that you don't need to know. So bottom line, rods are making inhibitory neurotransmitters, and light makes them stop doing that. Now, if you would like to ask questions about rods and cones, frequently, questions about color blindness will involve Unit 1B, genetics, and this unit at the same time, right? Because it's about the same topic. It's what makes it such a popular topic for diploma questions, because it's referring to two different units. Okay. Now, if this is super interesting to you, you might notice in this picture there's all these other types of cells back here that I'm not going to talk about. I took a bunch of neuropsychology classes in university, and one of them, actually a whole series of them, was about sensation and perception. And we went into vast amounts of detail about how all of the rest of that works. So if you're like, this is kind of cool, take sensory perception psychology classes. You might find it interesting. Now, aside from detecting light, your eye needs to be able to focus. The process of focusing is called accommodation. Accommodation is really referring to the ability of the lens to change shape. Now, when you guys dissected the eye and you took the lens out, it was pretty firm, right? And you might think, oh, there's no way that thing changes shape. But the lens is flexible enough that it can change shape, especially when the tissue is all still live and not preserved. And the idea is that the lens changes shape so we can focus images directly on the retina, right where the fovea is. So you'll see there's two sets of pictures and explanations here. On the left, we're taking a look at an eye from the side, and we're really only looking at one set of ligaments, one ciliary body. There would be some on the other side as well. Realistically, they go all the way around the lens. So this is just a portion that we're showing. This is the lens. These would be the ligaments that are attached to the ciliary body. If those ligaments pull tight, and they do that from both sides, so even though it's not in the picture, there would be some down here too. If I pull something tight from both sides, it kind of flattens. When that happens, it helps your eye focus rays from a distant object. Now, if you did or are doing physics, you get into all sorts of calculations with this kind of stuff, calculating focal length of lenses. We're not doing that. I just want you to know that the lens changes shape. Flatter lens is for farther stuff. Flatter, farther. See how they both start with F? If, instead of being tightened, those ligaments that are attached to the ciliary body are relaxed, so instead of pulling tight from both sides, I kind of let it go, the lens is more rounded. The lens is rounded because the ligaments are relaxed. So when it looks like that, your eye is better at focusing on objects that are closer to where you are. So flat is for farther, round is for closer. So if you, right now, look at your cell phone, which may or may not be on your lap or in your hand, or look at your paper and then look at the screen, your eyes just did this right now. So look down, look up, your eyes are doing it. One thing that you might be interested to know is that as you age, the ability of your lens to do this tends to decrease, which would explain why even people whose vision had been fine when they were younger might say need reading glasses because they have a hard time accommodating, focusing on something close as well as something far away. Does anyone want to ask anything about focusing and accommodation? I even noticed this happening myself. I'm like, I'm so young. How is this happening to me? I'm not. I'm fairly old, but maybe because I'm aware that it's happening, I look for it. I check these things. And so I've noticed that lately, especially at night, if I'm trying to look at something close, like a book, and then look up at the TV, there's like a lag of time before my eye adjusts to what I'm looking at. Now, most of that has been about sensation. How does light get to your retina? How does your retina tell your brain that it detected light? This last part is about visual interpretation. And I don't want you to get freaked out by this picture here, but I like the way it shows stuff crossing over, which is the point I was trying to illustrate. We have two eyes, and each of your eyes in combination with your brain will help compensate for your blind spot, which is why you had to cover up one of your eyes while you were checking for your blind spot. So because we have two eyes, they compensate for each other. So if you look at this top part of the picture, the green is showing what your left eye sees. The orange is showing what your right eye sees. And the yellow portion is the overlap. So most of what you look at, both of your eyes are looking at it. That's helpful because then you get two sets of information and your brain can compare those. Now the information from your eye, we know, leaves via the optic nerve. It's going to stop off at the thalamus and then go on to the cerebral cortex. So thalamus is a relay station. So make note of that structure. That's the part of the brain where eye information, visual information is going to go on its way to the occipital lobe. Now what's interesting is that the left optic tract, so what you are seeing with your left eye, your left optic tract, so anything that's on the left, is going to go to one side of your brain, but not the side that you would think. Left stuff goes to the right side of your brain. Right stuff goes to the left side of your brain. And that's true for most sensory and motor control functions. The right side of your brain makes the left side of your body move. The right side of your brain is detecting sensory information from the left side of your body. So if you look at the picture, see right here, it's crossing over. That happens with a lot of information, so that in the long run, when we get all the way back to the occipital lobe down here, the stuff that you were looking at on the left side is being interpreted on the right side of your brain. Now, like I said, we have binocular vision, which means we have two eyes. Bi means two. Both eyes are needed for us to see depth and for us to see 3 dimensions. With only one eye, you only get one point of reference, and it's really hard to see. The same thing will be true for hearing, and I'll mention my dad there because he lost hearing in one of his ears, and he said that it's really hard for him to tell where a sound is coming from because he's only getting information in one ear. And the same is true for vision. If you can only get information from one eye, it's harder to determine depth and 3 dimension, like figure out how far away an object is. Now, the occipital lobe simultaneously processes different aspects of sight. So what color is the light? How bright is the light? Was there no light at all? All of the information that your eyes are collecting through those photoreceptors is being processed here. So if I think about the pathway for interpretation, information is going to leave the eye through the optic nerve, go to the thalamus, then go to the occipital lobe. That's the pathway that information is following. That would make a good put stuff in order question, for sure. Does anyone want to ask anything about this? I would never ask you to redraw this picture. It was just so you could see the crossover. Okay. So if you want to test it out, it takes a little while, but if you cover up one of your eyes for like a while, you might notice how things look different. However, your brain is really good at compensating, and if you're looking at something that you had already been looking at and you have a reference, it might not look super different to you. It's got to be something like new and novel so your brain can't cheat. Now the last thing I want to mention are just some of the more common vision disorders. There are so many things that could go wrong with eyes and with vision. And I would not, and neither does the diploma, expect you to memorize all of them. Most of the time, whenever we're talking about a disorder on an exam question, the context box tells you what it is. Okay? I'm putting these ones here because these are the typical ones that get talked about the most. But as a general rule of thumb, anytime a disorder shows up, unless it was specifically mentioned in the curriculum, which it rarely is, diabetes is one of the only ones that is specifically mentioned in the curriculum, we would tell you what the disorder is. So one disorder that you may have heard of is glaucoma. That's a disorder that affects the aqueous humor. If a person has glaucoma, the aqueous humor cannot be evacuated. So your eye is supposed to be able to let that fluid drain and replenish. And so because the duct is blocked, your cells of the eye start to deteriorate because they're not getting enough oxygen. They're not getting what they need. Waste isn't being removed. The next one is a cataract. You tend to hear older people having this issue, although it's not exclusive to age. But because cataracts are when the lens becomes cloudy due to protein degeneration, it's usually associated with age. As we age, things start to degenerate. Astigmatism, the one that we just tested for right at the beginning, that is when your cornea is uneven and it makes vision slightly less sharp. It doesn't mean you can't see. So for example, some of you might be near or farsighted and also have astigmatism. Some of you might only have astigmatism. So you can still see, but it's not as sharp as it would be if it was corrected. Myopia and hyperopia, that is when people cannot see far or close objects. So myopia is when you have difficulty seeing distant objects. We would call this person nearsighted. They can see what's near but not what's far. In this scenario, the eyeball is too long. So eyes are supposed to be more or less circles, but in myopia, the eye is more football shaped. I might have exaggerated there, but I hope you get the idea. Instead of being round, it's kind of longer on the edges. The eyeball's too long. So when light is refracted by the lens, it falls on a spot that's before it even gets to the retina. It doesn't land directly on photoreceptors, which is what makes it blurry. Hyperopia is the opposite. So if a perfect eye is nice and circular, that's my nearsighted eye. Well, then a farsighted eye is like an eye that's too short. So it's not football shaped this way, but it's not wide enough. It doesn't go far enough back into the eye cavity. And so light rays, again, are not focused right on the retina. Now you can correct nearsightedness and farsightedness with glasses. Glasses essentially change the way light is bending so that it does land directly on your retina. If a person was getting laser eye surgery, we can't change the shape of your eye. That's not what laser eye surgery is. What they do instead is they reshape the cornea, which changes the way light refracts. And sometimes a person's eyesight isn't suitable for LASIK, When I got mine, I was right on the borderline of them not being able to correct it with LASIK. So you can't correct everything with laser eye surgery because we're limited by the size of the cornea. If they can't reshape the cornea, then it wouldn't work. And then the last one is color blindness, and we've already talked about that in the genetic sense. But color blindness is the inability to see certain colors, and it's when a person lacks one or more types of cones. And I showed you that plate of the Ishihara color test right at the beginning today.